So everything we're doing for the next few weeks can be found in chapter 17. So if you are looking for extra information, I of course am going to teach you things up here, but the textbook that you either have a physical copy of or that you're using online explains it slightly differently or might use slightly different examples, and sometimes just hearing a different explanation of the same thing can make a big difference. I'm also going to be posting a bunch of videos. A lot of them are from the Amoeba sisters. We've watched them before. We watched their DNA replication video that they made, like, it was a game and the characters were ligase and DNA polymerase and they were all little dudes. One had a tool belt. But they've made some really great videos on some of these genetics topics. And so we'll watch little bits and pieces of some of them, but I'm gonna post them more as an extra resource for you in case you're like, okay, I came to class and I did the homework, but I still need a little bit more explanation on what this topic is. So that'll be available for you. So our first lesson, I titled P's, Patterns, and Probabilities. And you'll see why it's about P's in just a minute. This is just a fun picture that I saw on the internet one day. I don't know if you can see because it's kind of small, but the dad has horizontal stripes and the mom has vertical stripes, and then the boy has a little crisscross pattern, and it says, genetics, this is how it works. And even though it's kind of a silly simplification, it kind of is. You get stuff from your mom and stuff from your dad, and the combination is the characteristic that you have. So, this little bit here is not something that I'm going to quiz you on, but I just want to give you an idea of the fact that we didn't always think that organisms got their characteristics from parents the way that we do now. If we go way back in time to Aristotle, Aristotle had a lot of really good ideas, and I'm sure you've heard about him before. I know I always talk about him in Science 10 when I'm talking about other things in chemistry. But Aristotle had some ideas about where organisms get their characteristics, but he also didn't understand cells and the way cells work. And maybe you remember learning about the cell theory and how cells have to come from other cells. And so if we fast forward a little bit, there's a couple of scientists, Van Leeuwenhoek, who you might remember as the microscope guy, and another scientist named René de Graaf. They both thought, respectively, that either the sperm or the egg contained the entire person, and that the other cell was just there as like a support cell. So for example, Van Leeuwenhoek thought that if you could zoom in on a sperm cell, in the head of the sperm, you would see a tiny little miniature person, like ready to go, in the sperm. You're supposed to laugh because it's kind of funny because we know that's not how it works now. But you gotta understand, before we had the tools to see things, that seemed like a very real possibility. Then someone else came along and said, no, it's the egg. The egg has the entire person inside of it, and the sperm is just the stimulus for the egg to grow, kind of based on the idea that the mother is the one who's producing the child, and so the egg must contain the entire person. Now, none of these are correct. This is not what we know now. We know that in organisms who reproduce using sexual reproduction, that the sperm and the egg are both contributing genetic material, but we didn't always know that. And so we didn't always know why organisms looked the way that they did. But we did, long, long ago, realize that if you want the offspring to look a particular way, you better find parents that have the characteristics that you want in the offspring. So even though we weren't quite sure how, people have been sure for quite a long time that offspring, children, babies, whatever, get their characteristics from their parents somehow. So this example down here is selective breeding, in particular with dogs, mostly because this is my, may she rest in peace, first dog that I ever had. Her name is Princess. She's a miniature Dachshund. She was the most adorable thing that ever lived on the face of the earth. Her legs were this long, and her stomach would touch the ground if there was a little bit of snow on it, so she wouldn't go outside in the winter. I would have to like pick her up and put her down so she could pee, and then pick her up and carry her back, because she was just such a princess about it. Anyways, that's not the point. But you know, or you probably have seen different breeds of dogs. They're all dogs, but the way that they look and the characteristics that they have are specific to their breed. And you might know that people breed organisms specifically for their characteristics. So my miniature Dachshund happened to have been a purebred miniature Dachshund. meaning both of her parents were miniature Dachshunds as well. So she came from a line of that exact species of dog. My new dog, who I don't have a picture of in here, but I should, her name is Lily. She's what I call shepherd's pie. She's definitely German Shepherd, you can see that, but I don't know what the other breeds are. She's some sort of mishmash, and she's a rescue, so we'll never know where she really came from. But she doesn't have as distinguishable of characteristics as, say, this dog does, where I could pick out her breed without even really trying. So we have known, even though we didn't know how, that organisms get characteristics from their parents. Now, selective breeding applies in other areas too. Do any of you raise horses or breed horses or know someone who does? Usually that's a pretty common thing around here. Oh, of course, with cattle as well. And so with a lot of animals in general that people keep either for as pets or as part of their business that they run, selective breeding is a very common procedure so that you can get, or is it dairy cattle that you're doing? Beef cattle. So of course you want the beefiest of the beef cattle so that you can sell your cattle and earn money that way. So the next thing we need to talk about is this guy named Mendel. He is a pretty important dude. He's what we call the father, or sometimes the grandfather of genetics. There's a photo of him here. You know what? Not bad looking fellow for his time. And he did a lot of experimentation with peas, which is why there's a joke about peas on here. So Mendel was a monk, and he lived in a monastery. And he had a lot of time on his hands, and he had a big garden where he was growing peas. And he started experimenting with these pea plants to see if he could predict or figure out how different characteristics were getting passed on. And so Mendel used what we call true or pure breeding plants. That is a term that you absolutely must know. A true or pure breeding plant is one, especially if we're talking about a plant, that was self-fertilized, so that both their alleles for a gene are the same. So an allele is one version of a gene. So for example, in his pea plants, one of the characteristics he studied, you can see up here, was what color the flower was. And so there was a gene that controlled flower color, and there were two different alleles, the purple allele and the white allele. And so what Mendel did was he would let the plants self-fertilize, usually over several generations, to ensure that they were not getting genetic variability, to ensure that if the plant had purple flowers, it's because they had two copies of the purple allele, and they didn't have two different parents. It was just the same plant reproducing over and over again. Now, in the beginning, Mendel did what we call mono, stands for one, hybrid cross. That is a cross based on a single trait. In other words, only one trait differed. And so he had a whole bunch of pea plants, a whole ton of them. And if he was trying to investigate flower color, he would make sure that all of the other traits were the same. And the other traits he studied were things like flower position, seed color, seed shape, pod shape for his pea plants. And he would make sure the only thing that was different was the color of the flower, so that he could be sure that that was the gene that he was studying. Now, he did a whole bunch of these crosses, and we'll talk about them as we go through some examples. But when talking about crosses, here are another couple of terms that you need to know. You need to know what a parental generation is. It will be represented with the letter P. The parental generation is always the true breeding plants that he started with. They're the first generation. So in whatever example we're discussing, whatever the first two plants or first two organisms we cross are, they will be the parental generation. The generation that comes after will be their descendants. It's called a filial generation. which is from another language, the word filial, you wouldn't say that a ton, but it's referring to the sons of. So we're trying to get across that we're talking about the children or the descendants of whoever the parental generation was. The first filial generation we would call F1. F for filial and 1 for the first filial generation. In Mendel's work, he would always take 2 of these F1 plants and then make another generation after that. So he would let these F1 plants self-fertilize and produce F2, a second filial generation. We could make F3, F4, F5, F1 million. Like, we could keep going forever and ever. But in most questions, maybe F2 is as far as we go, because otherwise it starts getting very big and difficult to predict. So frequently you'll see a parental generation, maybe 1 filial generation, and possibly a second one. And that's generally as far as we go. And you should know that we represent them with P, F1, and F2. Now, in terms of what Mendel studied, I want to be clear, at no point do you have to memorize his data. I would provide you with a table where you could look the data up, or I would tell you in the context of a question that we were doing what his data was. You don't have to memorize it. I've put this little picture here, so if you have the printed notes, you would have this out. There is also on page 588 a very similar looking picture where you could reference this data, so you don't have to have memorized what the flower colors were in Mendel's pea plants, or what the seed color was, or what the seed shape was, okay? So we're gonna do some examples with this data, but you don't have to have memorized it. Now, one of the things that Mendel discovered that's important for you to know is the idea of dominant and recessive alleles for a gene. The way that Mendel discovered what dominant and recessive were for a particular gene was he would do one of his crosses. So if you take a look, there's my P, standing for parental generation, like we saw in the last slide. And if you look at the 2 things I'm crossing, these are true or pure breeding plants. The one on the left has 2 capital R's, and I'll just write it a bit bigger here. This particular plant had a round seed shape. Now, Mendel wouldn't have known ahead of time that round was dominant, but the reason I'm writing the capital letter is because he discovered that it is. The plant on the right, over here, it says 2 little r's, and its shape was wrinkled. Now, the r's that I'm writing represent the 2 alleles that the plant has for that gene. The fact that they are both capital or both lowercase is because they are true breeding plants, meaning they have 2 of the same allele for that particular gene instead of 2 different ones. Now, what he discovered, Mendel, in the case of round or wrinkled peas, and those pea plants he was studying, was that if he took true breeding round plants and crossed them with true breeding wrinkled plants, the F1 generation that you can see here, 100% of them, all of them, were round. And so if we consider what that F1 generation should have inherited from the parent on the left, the round one, had to have passed on the round allele. That's the only allele it had. The parent on the right had to have passed on the wrinkled allele because it's the only one it had. So just so you can see a bit bigger, those offspring, the F1 generation, the first filial generation, they should all have 1 copy of the round allele and 1 copy of the wrinkled allele. And so you might think that they should look maybe round or maybe wrinkled or maybe somewhere in between. But what Mendel discovered was that all of the offspring, 100% of them, if he did this cross, always ended up with round seed shape instead of wrinkled. And so his first definition here is that a dominant allele is the trait that is expressed in the F1 generation. When we say expressed, we mean it's the trait that shows up. So if I take two true breeding plants and I cross them, and all of their offspring all have one trait and not the other, that trait that they have must be controlled by a dominant allele. And then conversely, a recessive allele would be represented by the trait that was not expressed, the one that the offspring didn't have. So in this case, round was expressed and wrinkled wasn't. That means round was the dominant allele and wrinkled was the recessive one. Now this particular example illustrates complete dominance, which is important because there are other types of dominance, but this first set of questions and set of lessons will focus on complete dominance. In complete dominance, it means that one allele completely masks the other. In other words, an individual with a recessive allele and a dominant allele, like our F1 generation, they have one round allele, one wrinkled allele, so one dominant and one recessive. So an individual with a recessive allele and a dominant allele has the same observable physical characteristics as an individual with two dominant alleles. What does that mean? It means my F1 generation looked round, just like my parent generation looked round. If I looked at those two pea plants, I wouldn't be able to visually tell the difference. I wouldn't know just by looking at them, oh, that one had two copies of the round allele and that one only had one. So in complete dominance, if you have one copy of the dominant allele, that is the trait that you see fully and completely, and you don't see the recessive trait. Now, would anyone like to ask any questions about that? Yeah, Caleb.
Can I just see a picture of that? I'm gonna print the notes.
You sure can. I'm recording this too, hey?
Oh, okay. I'll just do it later.
Yeah. Okay. Now, if we consider Mendel and his work with peas, he established a bunch of laws that you'll come to know as we go through the lessons. One of his laws is the law of segregation. And this law should seem familiar because it really has to do with the formation of gametes, and we already studied that. You know that each individual has two copies of every chromosome, right? That's what we study. Well, since genes are found on chromosomes, right? Genes are sections of DNA that code for a protein on a chromosome. They also have two copies of each gene. And these copies separate, in other words, there's segregation randomly, right? Randomly, because of independent assortment, during the formation of gametes, and each gamete receives a copy of each gene. Gametes don't get two copies of each gene, they only get one. Gametes are haploid. And so if you look over on the side, you can see I have a male and a female here. And I'm using the same scenario that I did on the last slide. This is a male and a female from that F1 generation from the last slide, where each of them had one copy of the dominant round allele, my capital R, and one copy of the recessive wrinkled allele, my little r. And so it's just showing you that if we consider their gametes, the male plant could make a gamete that has the dominant allele, or it could make a gamete that has the recessive allele. There are two possibilities. And the female could do the same thing. Don't worry too much about what's inside the picture yet. It's showing what their offspring could potentially look like, but we'll get there. So this law of segregation really is reminding you that you know what happens during meiosis, right? We start off with a diploid cell, then it goes through meiosis 1, becomes haploid, goes through meiosis 2, and those sister chromatids pull apart. Anyone want to ask anything here? Okay. So you have to be able to represent crosses And we will use Mendel as our example in the beginning, but we'll start looking at lots of other scenarios as well. And there are 4 terms that you need to be able to use. It's unlikely that I'm going to ask you on a test, what is the definition of a genotype? In fact, I promise that I won't. But I'm going to use the word, and I'm going to ask you, what is this organism's genotype? And if you don't know what the word means, you can't possibly answer my question. So a genotype is the combination of alleles that determine a given trait. So for example, with those P's I was looking at, I might say, well, the genotype for that F1 generation was capital R, little r. Those are the alleles that the organism has for that particular trait. Phenotype, same thing as genotype. I'm not going to ask you what is the definition of phenotype, but I might ask you, what is this organism's phenotype? So you need to know what the word means. Phenotype is the external expression of the character, so its physical form. So for example, me saying that the pea plant has a round shape, that's its phenotype. That's what it looks like. Now, if we take the genotype and are a little more specific with the possibilities, we could describe an organism's genotype as homozygous or heterozygous. Homozygous is an individual who has 2 identical alleles for a characteristic. So for example, our parental generation that we used in the example, where there were 2 copies of the round allele, the capital R, or 2 copies of the wrinkled allele, the lowercase r, those individuals have a homozygous genotype. A heterozygote, or the adjective would be heterozygous. And I like to change up the type of word that I use, sometimes the noun, sometimes the adjective, so that you're comfortable with that type of switch. I could call an organism a homozygote, or I could call their genotype homozygous. I'm talking about the same principle. I'm just using the word as an adjective in one case and a noun in the other. Heterozygote, or an organism with heterozygous genotype, is an individual who has 2 different alleles for a characteristic. So this big R, little r combination, that would be a heterozygous genotype. And you can't read the writing at the bottom, but I'll read it for you. Those are heterozygotes. Get it? Because they're goats. The front is black, the back is white, and underneath it says just allele uneven instead of just a little uneven. Oh, such a good biology pun. So good.